Welcome everyone. Thanks Margaret and Doug. My name's Stuart Denman. I'm with CSIRO in Brisbane, along with Chris McSweeney. We're coordinating the additives and supplements work area in NLMP II. I'm just going to give an overview around the product development and where we see the work area sort of sitting, and Chris will give more of an overview of where the different partners fit and the linkages and the facilities that they bring into the partnership. So I think we know that the products that we're looking at, as Doug and Karen have said, we've got ones that are nearer to market and there's ones that are more in the development phase. And all of the products, including the ones that are near market, have gone through that development phase where we've got data around the efficacy of the products, whether their mode of action is direct or indirect on inhibiting methanogenesis. And these subtleties are quite important in how we shape the research, particularly when we get closer to near to market. So inhibitors and additives that are going to have a direct effect on methanogenesis are more likely to be applicable across a broader range of the industry, and in fact could even be driven in towards other things later on around, or easily transferable into smaller ruminants, monogastrics, and even effluent management. But our main focus is going to be around beef in particular, and beef and dairy. I think the thing that we need to remember in all of this too is that these products have to have zero harm, and by that we mean not only in respect to any sort of impact on the animal, but we also need to make sure that these compounds are carbon neutral. There's no use in us producing products that are going to take an enormous amount of energy to make, because the whole idea of this is to keep a clean and green image for the livestock industry. So just a few rules are being, or a few measures are being across these products before we prioritise them down into the near to market sort of area. And so we're looking at things around the cost of production, the likelihood of adoption, and of course, more importantly for the industry, the scale of emission reduction and the productivity benefit back to the industry. Once we've sort of got those things under control, we see an initial phase 1 animal trial with some of these products that are in the development phase need to go first, where we're trying to get that data around dose response validation. And this data sort of feeds into methodologies, and that's usually around pen trials and controlled chambers. Once we get closer near to market where some of these products, particularly the 3-knot and Asparagopsis sit, we sort of split them into the intensive system and the extensive system. And these in themselves have different challenges that we need to address. Both of them are going to require delivery systems and measures that again look at methane reduction and productivity benefits. If we look at the intensive system, that's predominantly going to be around dairy and feedlots, where producers have day-on-day access to the animals, making delivery a much easier prospect. And this is probably why it's an easier or higher priority in the strategy for these product companies. So there seems to be a lot of focus on that, but the thing we need to remember that this is a low part of the sector, except in dairy, but the feedlot, at least in beef, is a low part considered to where most of it sits in the grazing system. So when we go to the extensive system, of course, we're going to have issues around delivery, and so questions and research around how we're going to deliver products and whether that will give us the same emission reductions and productivity gains that we're seeing in intensive or well-controlled systems are definitely questions that we need to answer. Now, these sort of strategies are not high on the radar, or they're not the first priority for these product companies, and this is probably where we need to be doing some research. And as I said before, although our focus is around beef and dairy in this case, we should also remember that some of these products are going to be able to be funneled into other agricultural methodologies, particularly around small ruminants and monogastrics and effluent management. So I think with that, I will pass over to Chris, who will give an overview of the linkages, and then we'll move on to the individual project leaders that will talk about their products.
Okay, thanks, Stuart. Yeah, my idea here is to just give you a little bit more detail about the product pipeline that we've devised for these treatments that are likely to come through the system, who's developing those products, and the facilities and equipment they have to undertake the work. And so what we've done here is just put together a product development pipeline, some of which Stuart has already explained, but this provides a little bit more detail. It's not prescriptive. Some products will go through a slightly different pathway, but in general, there's a lot of commonality in terms of how we think these products will progress through this system. So look, I'll just concentrate on the products themselves and who's producing them and where they map to this pipeline. So first of all, we have Queensland University of Technology and the University of Technology Sydney, and they're both looking at microbial or microalgal supplements and the development of those. And their products really are at a very early stage in this pipeline. They're doing some pre-evaluation of those products and at this stage, some very early in vitro assessment. Both these institutions have lab scale production facilities to develop these products. They're not reliant on, say, a commercial producer at this stage. If we then move further down, the University of Western Australia have a project which is looking at specific plant bioactives. They've already done quite a bit of in vitro assessment in that space. And CSIRO is also looking at screening in vitro a range of supplements or nutritive or supplements of nutritive value that would act as hydrogen sinks to redirect energy when animals are inhibited with methane mitigants. We then expect that the pipeline would progress through from that in vitro stage to fairly controlled animal health, respiration chamber studies, dose response work, and animal health evaluations. And in that particular space at the moment, the Sydney University are interested in evaluating conventional nutritional blocks, but with a range of additives, for example, tannins included in those blocks to produce reductions in methane. We think that those very controlled animal health studies are probably likely then to expand into pen trials, again with respiration chambers with more animals, not necessarily always going to that pathway, but quite often. And in this case, CSIRO have also proposed a project looking at nutritional supplements again to correct deficiencies in nutritional balances that would capture energy in the situation where we're reducing methane and hydrogen needs to be captured. These sort of trials under very controlled conditions with respiration chambers currently tend to be undertaken by University of New England, CSIRO, and University of Western Australia. They do have these respiration chambers. I may have missed somebody else who could have these facilities as well, but these particular institutions have significant resources in this space. As we then move from these control studies to the field, we need to consider that a lot of these products need to go from lab scale production to pilot scale production to provide the required amounts of material for these large numbers of animals. So on this particular stage of the pipeline, companies like DSM, who produce 3-NOP, have the capacity at this stage to produce tons of quantity of these materials, and Future Feeds are rapidly expanding their capability to produce large amounts again of the red algal supplement. QUT has pilot scale facilities for the production of their microbial treatments. But at this point here, we also need to bring in feed manufacturers who are also going to be needed to formulate blocks or other types of supplement delivery systems. Then, as we move across that barrier to near to market stages of the pipeline, where we actually move into the field, we're looking at this point again to maintain control over these products in terms of the development. We'd see that they tend to be used primarily on research stations, and in the field we then move from respiration chambers obviously to things like green feeds monitoring, which are more mobile devices that can be deployed into the field. And in that space, the University of New England and ourselves are proposing projects with 3-NOP and red algae. Coffee, we also believe, have actually got a project in that area, but we're yet to have the opportunity to have some more detailed discussions about their project. In that space, again, CSIRO, University of New England, Sydney University and the University of WA have equipment and facilities to do that monitoring in the field. Finally, we think then that these control studies and research stations need to then progress on farm to commercial scale operations, almost like producer demonstration sites where we can get herd scale productivity responses, actual measurements and variability of those reductions on farm for methodology development and some cost benefit analysis. And at that stage, we really need to engage more broadly with industry to bring on commercial partners who are willing to work with us at that herd scale. And we think that is the preliminary step then to market. Okay, I'll leave it at that point, and I think we're now going to hear in some more detail about these individual projects.
Thanks, Chris. It's just Margaret. That was a really great overview, Stuart and Chris. And I just wanted to say that we've got a lot of projects in this work area to get through in about 20 minutes, actually. So I'm afraid I'm gonna crack the whip on 2 minutes a project. I just wanted to apologise in advance for that, just in the interest of time. Thanks, everyone.
Yeah, thanks, Margaret, and thanks, Chris and Stuart, for a great overview of the area there. So the product that we're looking at in this project is yeast that have been engineered using biotechnology to produce known or new methane inhibitors. So known inhibitors might be bromoform, or the new methane inhibitors might be some natural products from plants, or even advanced biotech products which are used for silencing of genes to inhibit the pathways that produce methane. One of the advantages of this approach is that yeasts are already produced at a very large scale as protein supplements for feed, so the manufacturing is known and can be cheap and can be scalable. What we're really adding here is using the same yeasts, but adding in the products which have this efficacy for reducing methane. So it's really a value add on an existing manufacturing process. We've thought a little bit about the pathway and how we need to work with this. So the development work and so producing the yeasts and engineering the yeasts to make these products and to quantify and measure how much of these products that they make will be a close collaboration with Macquarie University and University of Queensland and Queensland University of Technology. And then we'll work with companies and really we hope to integrate with other university partners, or in fact we need to, for the livestock studies. So this is a very collaborative project going forward. And then going forward for commercialization, we're currently in negotiation with the companies, but we want to work with companies who work at the feed level and who are used to selling feed products, but also ones which have got the experience in that manufacturing of yeast, and also companies who are deeply engaged with the regulatory pathways so that we can address the approval of these products and get them into market. So the second project that really aligns to this, and again will be very collaborative, is the recognition that we do need to be able to scale up, produce and formulate any microbial feed supplement that is going to be made and fed to livestock. So these need to be formulated in a way which is stable so that they can be stored, transported, before they're used, and they retain their efficacy. And they need to also be able to work in a variety of different feeding systems, either in feedlots or extensive grazing. And they also, for any of the products that are microbial, need to be produced in sufficient amounts that we can do large-scale feeding trials, and that really needs to be included and very much a part of the project. So to do this, as Chris and Stuart mentioned, QT does have a lot of experience in this kind of lab to pilot type work. We've done feed supplement work before, where we've gone through from lab scale development all through to pilot scale and large scale feeding trials with partners. So we have infrastructure such as the QT Mackay pilot plant for doing this type of work. What we want to do as well is to say, look at the effective formulation, so that when the yeasts or other microbial products are dried to a powder, that they retain their efficacy and activity. They may also be encapsulated or put into lick blocks, for example. When we do this type of work, we always include techno-economic models pretty much in the start of the project to make sure that they're targeting the right areas for improvement, and also align to that, work with engineering design so that we have processes which are ready for tech transfer to manufacturing companies. So we are looking, as well as the products we might have mentioned in the first slide there, we're definitely looking for people to approach us for collaborative opportunities for other supplements that we can integrate into this project, and ultimately be in a position where we can deliver these materials at large scale in a proven economic system that accelerates the path to market. So thank you very much, everybody.
Thanks, everyone. My name's Peter Ralph. I'm from the University of Technology, and Janice McCauley's here with us. Our project is linked with Young Henry's Brewery, and I wanted to take Stuart's point that he raised earlier about the carbon neutrality of the products. So where we started was working with the brewery, and we're capturing the carbon from the fermentation of the brewery to make the microalgal products. So this project is about microalgae, not seaweed or macroalgae, but we're looking along the same paths of reducing methane production. So the product that we're working with is brewer's spent grain as a feed supplement, but putting the microalgal supplement into that product and having the reduction in methane. So some of the endpoints that we're measuring at the moment is total gas and methane production, the volatile fatty acids, microbial consortia, and how we have to pre-process the microalgal product to get the maximum efficacy. Next slide. So what we're doing is we're running a range of different analyses, in vitro ruminal assays. So we're running both manual and automated systems for looking at the methane potential. We're working with Alex Shivers from Sydney Uni, getting access to ruminal fluids. The endpoints that we're measuring there are proximal analysis, so we're looking at total moisture, the dry matter, the organic matter, and the ash-free weight. So the analysis that we're doing also involves techno-economic analysis, so we can go back to the process to ensure that we're optimizing the engineering and making sure that we're having the maximum gains for where we're putting our energies. So far, we've tested 5 out of the 20 species that we proposed to, and we are getting evidence of methane reduction in the in vitro trials. The next phase that we're looking to move towards will be both with Alex, some simulated ruminant analyses, as well as going to animal trials. And so I think this is going to be part of this discussion, is how we can work with partners to go forward. We have got some algal producers ready at scale, but I think the fact that we're working with a brewery, it means that these products can be, as I said at the beginning, carbon neutral to start with, and then reduce greenhouse gases through the methane reduction. Thank you.
Okay, hello, good morning, everyone. I'm Luciano Gonzalez, and I work for the University of Sydney. This is a line just showing the research team that we have put together to work in this project. So if we can go to the next slide, please. Beautiful. So we've been doing a lot of research with leak blocks and changing formulation and different additives in feed blocks or molasses blocks. The objective of this project is to develop practical, economical, and effective solutions to reduce greenhouse gas emissions, but also to improve productivity, particularly in the rangelands. We have research projects going in Northern Australia to Southern Australia. What we want to do is expand this work and do field work and pen trials with the leak blocks. We have the green feed measuring systems, and we also have electronic feeders, walkover weighing, and supplement monitoring systemes for grazing conditions. And what we want to do is to try different combinations of different anti-methanogenic additives. Also work on the feed block design to control the amount of consumption of the animals, like for example the hardness of the blocks or sweetness and salt content. And we also want to look at block structure, what we add in there to improve the efficacy of the different products that we are trying. The other things that we have been doing and we want to test as well is all the delivery methods, including pellets and salt, loose salt or loose leak as well, and look at the dose response, how we can increase and optimize the response in a way that we balance reduction in greenhouse gas emissions and productivity. We have done some in vitro trials already. We have seen reductions on the amount of methane produced per gram of organic matter of up to 3-fold. So we want to continue that work and now move into more pen trials with the green feed systems, and also to in vivo trials, from small trials to large scale pastoral trials in Northern Australia particularly. So that's it, everyone from us. Thank you.
Okay, I'm next. So Zoe Dorris from UWA. So our project is on plant-derived feed additives, in particular for extensive livestock production systems. So in previous work with MLA, we actually found some plant compounds from essential oils that can reduce methane production up to 70%, and without adversely affecting fermentation. And this was done in Rositec. So the next step we are planning to do is to confirm these findings in vivo, but also to develop some practical and cost-effective delivery systems for extensive industries. So we partnered up with Ridley, and so we are proposing to have a look at in vivo, in particular to acceptance, dose, extent, and persistency of methane abatement, but also any other benefits that we might achieve with these supplements, and also adverse effects that they might have. The second aim is looking at delivery systems, whether it will be drench, leak, block, or slow release capsules or micro encapsulation. As you can see, that there is a standard, as Chris was saying, standard way of testing things. So we have Rositec system where we will be selecting treatments that could be promising to proceed in vitro, in vivo. We'll be confirming effect in the chambers, and as you can see, we have the sheep methane tents or chambers, and then we'll test these in on-farm grazing studies. We have access to these butcher boxes in WA. Can we go to next slide? In terms of adoption, commensurate pathways to adoption, so these compounds are pure compounds, just to clarify. So these compounds are pure, of high purity. They're known structure, and they are actually readily available in commercial quantities, so you can purchase them. So we don't, at the moment, don't have to worry about sourcing them or making them. They are naturally occurring compounds in wide variety of plants. So we don't expect that there will be any limits, or there is no registration needed. So there is already work and registered products all around the world in Europe and US that are based on similar compounds. And
Hi Zoe, it's Margaret. Might just have to try and wrap up if you don't mind.
Yeah, sure. So what we need to do is just verify under control conditions. And we have commercial feed operator that will help us formulate the diets. Some possible constraints, you can see here, dose might be high. We might have issues with acceptance, cost, availability, and also adaptation, whether we need to provide all year round or both. In terms of, testing, we don't have access to green feed, and also, as you see, we are planning to do all our testing in sheep. However, if there is interest to be tested in cattle, again, we're open for collaboration on that.
Okay, sorry, it's Mary Fletcher here from the University of Queensland. We're actually talking about a project that's a collaborative project across with various parts of UQ, from chemical engineering to School of Agriculture and Food Science and to the vet school plus QAFI, but also involving our collaborative partners with DAF and some industry contributions. It's a large team, and there is some sensitivity about the IP, which is why we'd ask for a confidentiality agreement before we discuss too widely. In brief, I guess we're looking at a slow release system of bioactives, particularly 3-NOP in this instance. We've had discussions with DSM, and they're interested in what we want to do. So we're looking at in vitro fermented trials, in fistulated animals, long-term trials in rangeland cattle, and particularly looking at the impacts on the microbiome, methane production, and of course animal production and weight gain. So one component of the trial of the system is looking at young calves to study the effects on the establishment of the microbiome. So clearly we've got access to green feed, although we intend to purchase more. We've obviously DAF's an important partner in this with the research stations and the access to cattle, and also the microbiome work. And we intend to feed the economics of this into with the bioeconomists in DAF to validate the actual impacts from an economic point of view, as well as the animal production. So that's about a summary of where we're at the moment.
Just to add to that, if I may, Margaret, this is Ben Hayes coming in here. Mary's also using this technology to mitigate the effect of plant toxins, and that technology has been very successful, which is why we're very excited about applying it for this purpose. So thanks, Mary.
Thanks.
Okay, thanks for the opportunity. Roger Hegarty here. Look, these slides probably aren't the most appropriate given what other people have showed. I thought we were meant to show competencies and project interests rather than details. But to put it simply, on UNE campus in Armidale, where we work closely with New South Wales DPI and share facilities, well equipped with respiration chambers and green feed units for both sheep and cattle. We do in vitros the same as everyone else, batch and Rusitex. But I guess we have the research feedlot, and we can see our role in NLMP. If I can go down to the next slide, thanks. Is really to marry up two areas of work. One is the breeding component that Paul Arthur will lead and talk of, and the supplement work. and we can see applying those tools like 10 green feeds for 6 months of the year in the genetic evaluation in the feedlot. And then particularly we see NLP as an opportunity to set up a replicated managed grazing system where we can use walkover weighers and auto drafts and green feeds to get live weight change as well as emissions from animals on 3 or 4 different supplements at a time in the grazing trial. And if we want to, we could take them through basically to get a whole of life stuff as they finish in the feedlot. Now, what compounds are involved there is you know, we're not too worried about. We work with MLA and CSIRO and DSM at the moment with 3NOP, just starting some dose response and productivity stuff with Future Feed. Work strongly with Olson's and their lick blocks. So there's a range of things that all have this same need to say in the grazing environment, what's their productivity response and what emissions mitigation do we see in the paddock? So we really see, I'll probably get shot if I say it as an infrastructure, but we really see that what the industry needs is a tool to get that on-farm productivity and mitigation data. And so we're trying to set that up in a structured fashion right next to our feedlot where we can get it in a replicated, highly publishable, something that's really good for supporting methodology developments. And we'll apply that with as many products as we can in the life of the project and as many partners as possible. And that's probably all I need to say, but happy to talk later.
Okay, so Chris McSweeney back again. What I've done here, I've just got one slide. We've got several projects under development, and I've just broken them down into 2 broad areas. Northern Australian rangelands, where we are planning to evaluate the 2 lead products, 3-NOP and red algae, under extensive grazing systems. And this is really a collaborative project with the University of New England, and in particular, Roger Hegarty, who's going to focus more on southern Australian grazing systems. Within that rangeland application, we've got 2 approaches. One is early life intervention, with some of these products to intervene in the early life calf to establish a low methane rumen in terms of the microbiota, but also to deliver these products to adult animals in the field, looking at the mitigation potential and the productivity responses to the products over the annual life cycle. So taking into account seasonal changes and also pasture quality differences throughout the year. The other area where we're focusing on is this notion of improved productivity with capturing the energy that's normally lost as methane. See this is essential to getting adoption, but needs really some fairly early stage basic work. So we're looking at that in terms of capturing the lost energy in the form of the carbon or hydrogen that's associated with methane. To do that, we're looking at screening feed additives that would be of nutritional value in their own right, but also acting as hydrogen sinks. But also we think there's opportunities to correct nutritional imbalances in the diet, particularly in the northern system, which then would feed into maximising the energy capture associated with the reductions in methane and accumulations of hydrogen. Okay, thank you.